You are doing well.
Hello,
Hello.
how are you?
Hello, Daniel. How are you doing?
I'm good.
Good. Glad to hear from you, man. Okay, the main purpose for our meeting today, it's like a review, not very, very much a review, but I need to focus mainly on the last two chapters. The final basically is going to be those two chapters, which is DNA replication and gene expression mainly. And then, because the final is supposed to be like cumulative, as you know, so there will be some selected chapters. I'm not going to select too much, but as a matter of fact, I'm going to focus mainly on the last two chapters, the Mendelian genetics part. Cell cycle again, I need to focus on this, and probably a couple of chapters, previous chapters, cell respiration and photosynthesis basically. But I'm going to send you like an announcement, more details about what you expect to see in your final. And as you know, the final exam is going to be online again and next week, and the date need to be determined. So this week you need to focus mainly on the lab practical because you have lab practicals this week. Mine, for example, is going to be open and available starting Wednesday till Friday this week. And then next week we're going to have the final for the lecture. So before I start here, it will be more efficient if you have any question for me before we start. Do you have any question for me before we start? Anything that need to be clarified? Anybody?
No, I don't have anything specific.
Okay. Okay, so anything mainly in any lecture like those, especially those two lectures that I send you, the DNA replication and the gene expression, I will focus on the gene expression one. I'm going to share with you the presentation. I need to talk about something very important, especially when I give you like a piece of DNA and ask you to do transcription to create a messenger RNA and then follow up with translation. So I need to make sure that when you have a piece of DNA or some DNA sequence, how to deal with it, which strand you are going to select for transcription, and how you translate that strand, how you translate the messenger RNA and make protein out of it. I'm going to show you the table, how to read the table and how to convert nucleotides to amino acids. This is what I'm going mainly to focus on because I think this is very important for you to know, because based on the last pub quiz that I gave and based on the answers that I received so far, it seems likely that nobody get it right. Okay. So let me do this first. I need this pop quiz 11. Let me share it with you because you do this. Okay, now on the screen, can you see pop quiz 11?
Yeah.
Yeah.
Okay, great. So when I look to this one here, you did good job. As a matter of fact, everybody did get very good job. One of the questions that was kind of hard a little bit, I believe, was the okay. For those, question 1 all the way to number 8, okay? Do you remember which one of those was kind of hard or difficult for you, or you didn't get? When you say about which of the following is not a reason that peas were well suited for Mendel breeding experiment, one of those is not true reason. Or not a reason as at all that why Mendel selected peas. Do you remember the answer for this? All those considered to be advantage except for this. When you say peas have unusually long generation time, this is wrong, because no way, because he selected peas in general because their generation time is short. Okay. When you say a pea plant is heterozygous, and you need now to know what is the difference between homozygous and heterozygous. So a heterozygous, when you have the two alleles are different, okay? Like in this case here, when you say heterozygous, like you have two alleles, they are not the same. If they are the same, meaning like P capital, this is homozygous. If it is P small, this is homozygous. But when you have two different alleles, so it's heterozygous here. So it's a plant like heterozygous for two things, for the color and for the seed. What do you think about the type of gametes that can be produced from this plant? So remember, gametes are going to be haploid, okay? So meaning that each gamete will have only one allele, okay? So if you think about these two gametes, first of all, wrong, because you are expecting to see 4 gametes. Remember, meiosis produce 4 cells. So when you look at this answer here, you need to basically eliminate those. That doesn't make sense. So you have 2 gametes, this doesn't make sense. You need to get those out of the picture. Now you have 4 gametes, 2 questions for 4 gametes, 2 answers, I mean, for 4 gametes. So which one you will select if you think about, the possible genotype for the gametes, okay? So you have P capital, Y capital. You have P capital, Y small. Okay, so if you look here, you have P capital, 1, 2, 3, P capital, Y capital. Can you see any P capital, Y capital here? No, right? Because you have there's a possibility that allele P, capital P, will go with capital Y allele. If you look here, you will not be able to see this. So again, this is the wrong answer. So the right answer is going to be this one here, okay? In this one, you say a cross between homozygous purple flower and homozygous white. So meaning that this is pure, okay? Alleles are the same, not heterozygous. So the offspring is going to be purple flower, mainly. This is what he did. So what we call this? We call this like true breeding, or we call this complete dominance or co-dominance, or the blending model. First of all, there is no blending model. There is no co-dominance here. So now what do you think about this?
That's a complete.
Huh? Yes, absolutely complete dominance between this, okay? And here is a genetic counselor is working with a couple who have just had a child, and the child is having a disease, okay? The thing is, parents they don't have the phenotype. So they found only that the kid or the child have the phenotype, okay? So they don't have the genotype. Even their family, nobody showed the phenotype. So that was weird, right? So now they need to ask the counselor, what's happening? We are okay, we are healthy, we are not diseased, our family are good, in good shape, but our kid has a disease. So what do you think the counselor will tell them? to explain this. First of all, he will not be able to tell them that you are not really going to see another baby. He cannot expect this. Okay, so this is wrong. He said, because you have had one child with these symptoms here, or this disorder, you must each carry the allele. And child you have has a 50% of having the disease. Look at the disease here, okay? The disease is basically, it looks like, because nobody else had it, so there is no possibility that they will have the 50% chance here. Because you have had one child with shy, you must each carry the allele, and any child you have has 25% chance. Okay, that is the right one here, because this is like a recessive trait, and it looks like both parents are carrier. So there is every single pregnancy, there will be a 25% that the child will have the disease. Okay. He can't say that. When you look at this kind of question here, when you say 25%, it doesn't mean that if you get if they got the first kid with the disease, that means that the following pregnancy is not going to be safe. No, it is every single pregnancy there is a 25% that the kid will have the baby will be like having a disease. Okay, so that will be the right answer here. This one, I think I have for this one, for 5 and 6, I have a PowerPoint I'd like to share with you to go through this in more details. Here, let me switch to a different screen. John is 47, has just been diagnosed with this disorder here, Huntington disease. When you have this kind of question, you need to read very clearly and try to get as much information as you can. So first of all, this is a disease which is rare and caused by a dominant allele. You know what is a dominant allele? A dominant allele meaning that one allele is enough to show up the trait. Okay? Now it says his daughter, which is 25 years old, and her son, which is 2 years old. He has a daughter and his daughter has a son. And then it says here, no one else in the family has the disease. When it says no one else in the family has the disease, that means that John is not homozygous for the trait. Because if John is homozygous for the trait, every single kid will have the disease. But since no one else in the family has a disease, the wife or the kids, meaning that John is the only diseased here and he has only one allele. So you look at the genotype for John, if we donate for Huntington disease with an H letter, so it's capital H here, dominant, small h is recessive. So John is heterozygous dominant, okay? His wife is homozygous and she don't have any disease. So when you look at this kind of genotype for John and his wife, so are going to expect that his daughter is, or any kid, he has, every time his wife is going to be pregnant, there is a 50% chance that they will be diseased. Why? Because that allele can be transferred. Like if you have 2 sperm here, one with capital H, that will fertilize an small egg. This is a person with the disease. If this is another sperm, we'll go with this one. This is another again 50%. So the whole thing is going to be 50%. So what is the probability that the daughter will develop the disease? That would be 50% chance based on this kind of like equation, and based on this kind of information. So again, when I gave you this kind of question in the test, try very much to extract as much information as you can. And because based on the information, you will be able to find the genotype. Because if I say that every single kid John has is basically showing the disease, that means that John's genotype is going to be homozygous, not like this. Do you have any question for this one here?
No.
There is another one here, blood typing, okay? ABO blood typing in human. First of all, when it says codominance, meaning that there are the two alleles will show up the phenotype. So there is no one allele is dominant over the other. So both will be the same, codominant, will be expressed same exactly. No one is going to be hidden. No gene will be hidden from the other. Both genes will be expressed. When you say what is the likelihood of a type A father and type A mother having a type O child? So first of all, you need to know this. When I say type A father and mother, there are possibility here that the father will be either homozygous, meaning pure, so the two alleles code for the blood type A, or it will be heterozygous, okay? So meaning that it has one allele for A and the other one which is a recessive allele that can give an O. So now this is three possibility here. So the first possibility that both the dad and the mother, they are heterozygous. If it is the case, so there is a chance that every single pregnancy, the baby will be 25% of the pregnancy, the percentage will be type O based on this. Okay, so you will see O blood type if both parents are heterozygous in this case. What if both parents are homozygous? They don't have the I small allele for blood type O. So they will never have any kid with blood type O. So the possibility to have blood type O is 0. There is a third possibility where one of the parent is homozygous, okay, type A, and the other parent is heterozygous. Same deal here. If you look at the babies, there will be a 0 chance for any baby to be type O, because for a baby to have type O, or for a child to have type O blood, he should have this this allele, I small allele, and that will get this only from heterozygous parents. Okay. Is it clear? Please help me and ask me if you have any questions. So I will make things more clear if I need to do that. Anybody has any question in this part?
No.
That's clear too. Anybody?
No.
Okay, let me move to it. Here, this is kind of more complicated kind of question. There is a cross, and this is the genotypes here. And if you have this kind of genotypes, it will be impossible to make, you imagine making like a Punnett square, how many boxes you need to create in that Punnett square to be able to figure out like an offspring with this genotype. It will be tedious. It will be too much work to establish like a Punnett square. If you are talking about like how many uh, uh, gene involved in this process, 1, 2, 3, 4 genes. That would be hard to do. So if I ask you this and I give you the genotypes for parents, and I tell you what is the likelihood of having an offspring with this particular genotype, you don't need to do Punnett square, because that will be too much work. So it is very simple. It's based on the probability here. So you put the genotypes for both parents, as I put here, and then you put the genotype for the offspring that you are looking for, okay? And then you take each gene, one gene at a time, and see the possibility that the genotype will show up. So you are looking for AA genotype. So now, you go ahead and start with this A capital, A small, A small, A small. So the possibility to have an offspring with the genotype A capital, A small is going to be 50% or half, because you have A capital, A small, this is okay. A capital, A small, this is another one. And then A small, this is not going to work. A small, you are going to neglect this. So what is left? You left 50% is going to be A capital, A small, so which is half. And then you shift to the second genotype, P small, P small. So again, you take the genotype for the parents and try to see the possibility of getting those gametes together. P small is going to be 25% or fourth. Do the same for the third gene, E capital, E capital. What is the chance to get those two from across like this? Again, that will be another 25%, because E capital, this is okay, but E capital, E small, no. E small, E capital, no. E small, no. So we have only 25% to show E capital, again, which is fourth. Finally, what is the possibility to get this genotype here? H capital, H small, from this kind of cross, it's going to be 100%, because H capital, H small, H capital, H small, H capital, H small, H capital, H small. So this is 100%, which we represented with 1. So if you multiply all those together, simple math, you are going to end up with this ratio here. So the possibility of getting this genotype from this cross will be 1 in 32. Did you get that one?
Kind of.
Yeah, well, yeah, ask me. This is the main purpose of this. You ask me, you know, how you get it. So as I
Can you
mentioned, when I get
explain?
to this kind of question again, you put the parents' genotype, and then this is the offspring that you are expecting coming from this. So if you do this in a Punnett square, as I mentioned, that will be huge square. It will be very confusing and very exhausting. So there is a simple way, called the probability way, that you can do that. Now, you look at the genotype of the offspring and you try to compare it with the genotype of the parents. You take one gene at a time, okay? So what is the possibility of having these two alleles together? The A capital allele from one parent and the A small allele from the other parent. So you take those two together and see here. The possibility to get an A capital, A small is going to be, see here, this is A capital, A small. We need this. This is one possibility. Is another A capital, A small. This is another possibility. Though now you have two, okay? 2 out of 4, remember. A small, no, because we can't. A small, no. So what we have left, 2 out of 4, which is 50% or half. And you keep doing the same for every single gene. and then you multiply, and you will be able to get the possibility or the probability of getting this kind of genotype from this cross.
How did you get two lower cases of the B's?
Which one?
The B. How are they both lowercase?
Oh, you mean this lowercase? Okay, now
Mhm.
take this. This is a heterozygous capital B, small b, capital B, small b, right? So in order to get B small, look here. The first probability, B capital with B capital, it is not going to work. It is out, one out. B capital, B small, again, no. That will be out. B small, B capital, no. That will be also out. Now, B small with B small, yes. So now we have 1 out of 4 that we are going to receive this.
So why weren't the E's both lowercase?
Which one?
The E?
E?
Yeah, why was it capital?
No, because this is a question here. If you look at the question, it tells
Oh,
you,
okay, I see.
yeah,
I see.
this is what you need. It tells you, I need this particular genotype.
Okay, I understand now.
Yeah, so based on this particular genotype, you put it here and you try to get it from the cross.
Okay, I
Yeah,
understand.
so this is the one that is going to direct you, you know what to select. And you are going to select from the parents' genotypes based on this. Okay. Any question for me? So let me finish with this and go back now to the main purpose of this meeting. I don't know how to do this, man. Stop share, and then I'm going to share a different screen now, okay? So I am going to, which one is the gene expression? Let me share this. Okay, let me go, you know, this is the gene expression chapter. Can you see this this PowerPoint presentation now?
Yes.
Yeah.
Okay, great. This is mainly for gene expression. Let me start to review with you first and see your information here, and if you was able to like comprehend the information in the PowerPoint presentation. Okay, so here is a question for you. In the process of producing messenger RNA, before I start, remember in biology, there are two processes for gene expression: to convert genotype to phenotype, to convert the gene that you have to eye color or hair color. There is two processes. One of them, we call it transcription. Transcription meaning a messenger RNA is going to be copied from the DNA. And the other process, we call it translation. And this is where protein will be formed from the messenger RNA, okay? So those two processes in biology, they are very, very, very important, and you need to be able to comprehend and understand those. When I say in the process of producing messenger RNA, okay, what molecule act as a template when we produce messenger RNA? So what is the template here? Meaning the main, the thing that the enzyme will work on to produce messenger RNA. So messenger RNA comes from what? What do you think? Is it coming from protein
DNA.
or from polysaccharides or from DNA? Yeah, I need some, like, we're going to discuss this together. So I'm going to ask you a question, and whoever knows the answer, you tell me the answer.
It's a DNA.
Yes, so the DNA is going to be the template for messenger RNA, okay? In this process, or what is the name of this process where RNA or messenger RNA is formed from DNA? Again, remember, we call this transcription. So never forget this, a transcription making RNA from DNA. What the enzyme catalyzes this process? This process making RNA from DNA needs some catalyst. need enzymes. So what do you think the enzyme here that will be responsible for this? Is it going to be a DNA polymerase or an RNA polymerase or a primase or a helicase?
It's the RNA polymerase.
Yes, RNA polymerase, because this is the enzyme from the name that make RNA polymer. RNA polymer, so an enzyme that make RNA polymer, we call it RNA polymerase. Remember, it ends with an ase here. So all those are enzymes, but this is the one which is responsible for making RNA from DNA. Okay, based on the other, like enzymes, can you tell me the function of each one of those? DNA polymerase, for example, what is the function of DNA polymerase?
Doesn't it catalyze the DNA replication?
Absolutely, yes. Again, like RNA polymerase, which make RNA, DNA polymerase make DNA, and we make DNA during cell cycle, during the replication process, during the S phase, where DNA is replicated. Who's replicating the DNA is an enzyme, we call it DNA polymerase. What about primase? Again, during the process of DNA replication, DNA polymerase cannot start making new DNA unless there is a short nucleotide or short oligos. We call it primer, should be there. And the enzyme that make those primers, we call it primase. So DNA polymerase cannot do the job without having something to build on. And that something, we call it primer. And there is an enzyme called primase to make that primer. So the process can start. What about helicase? Again, during DNA replication, DNA is double helix and it is intertwined, right? So you need to like unwrap the DNA so all the machinery for replication will have an access. So that enzyme here, helicase, open this double helix, opens the structure so the enzyme like DNA polymerase and primase will work to make new strands. All these kind of things you need to understand those, because your test, roughly, I can see 60% of it or 65% will be 2 chapters, DNA replication and gene expression. And the other 35 or 40% will be some selected chapters. I will tell you exactly what to focus on for the final. Here, remember for the when I say RNA polymerase, for the RNA polymerase to start making messenger RNA, there is a specific region in the DNA that the enzyme bind and start the process of transcription. Do you know the name of that region? So the RNA polymerase is not going to bind randomly. There is a specific region in the DNA where the enzyme binds. And we call this region the promoter. So what is a promoter? A promoter is a DNA sequence where RNA polymerase binds to start transcription. The direction and the polarity of the making RNA always DNA replication or transcription, always going from 5' end to 3' end, always 5 to 3. This is the 3' end. This is where the hydroxyl group is attached. And this is where new nucleotides are added to this. So remember that direction of transcription from 5' to 3'. This is very important because if I give you a piece of DNA, double-stranded DNA in your test, and I ask you to do transcription for me, you need to look for the strand of the DNA which says 5' to 3'. Okay, that strand that says, sorry, 3' to 5'. This is your template. That will be your template. Okay, why this is a template? Because the new strand or new messenger RNA is going to be formed from 5 to 3. So it should be anti-parallel to the strand. We'll go to this later. But remember, always the growing of the DNA or RNA from 5' to 3', because new nucleotides are always added at the 3' end. Okay. Here, in the process of producing proteins, now this we call this translation. Which molecules serve as a template? So the template for this process is going to be your messenger RNA. So messenger RNA is a template to make proteins. And this is, I will ask you this. Name the organelle that is responsible for assembling and making proteins. Can you tell me the name of the big guy who's doing this process during translation?
The ribosome?
Absolutely, yes, perfect. Ribosome, those without ribosomes, the process of translation and making proteins is not going to happen. Here, you need to know that ribosomes between bacteria and like eukaryotes, like human, for example, they are completely different. Structurally, our ribosomes are different from bacterial ribosomes. This is why a lot of antibiotics that we are taking are very selective for bacteria, because they target bacterial ribosomes, not our. So based on the structural differences between bacterial ribosome and the human ribosomes, lots of antibiotics are made, and those antibiotics are selective, so they kill only bacteria and they don't kill us because they select for bacterial ribosomes. Let me ask you this. What do you think about viruses? Do you think viruses have ribosomes?
No.
No, they don't have ribosomes. Our viruses do transcription and translation.
No.
So how they live, how they multiply and increase in number? They do, as a matter of
With the
fact,
bacteria, but how?
huh?
It's through the bacteria, right?
I'm talking about viruses in general. Do you think viruses have ribosomes?
No, I don't think so.
Yeah, they don't have ribosomes. So how they make their protein? How a virus like corona, for example, make all these virions, one virus particle enters the host cell and hundreds comes out. And in order for a virion to form, it needs proteins, like say protein coat and the capsid, all the spikes that you can see on the virus surface, those are proteins. So how the virus make those? Is it protein, basically cell like host protein that the virus steal in its way out, or those are specific viral protein? Those are specific viral proteins. So how virus make those if there is no ribosomes?
Don't they change the DNA of like the host cell to make it?
What they do, they use the translation machinery of the host to do it for them, basically. So it's kind of hijack of the host and direct all metabolic activity inside the cell for the virus to multiply and increase in number. So, and this is interestingly, the viral RNA or messenger RNA is very, very, very similar to eukaryotes RNA, not to bacteria, because it needs to like deceive the cell, the host, and tell the cell that this is your RNA, this is not a foreign RNA. So the cell will blindly start making protein, thinking that this is an RNA for host purpose. But as a matter of fact, it's an RNA that comes from a virus. So the virus RNA uses the machinery of translation of the host to make proteins. Okay, let me ask you here. For the ribosome to start like making protein on the messenger RNA, there is a difference between bacteria and eukaryotes. If you come with this, if you answer this for me, I will give you 10 points on spot. 10 points, whoever will come with the answer. Now, ribosomes will start the process of translation, making RNA, but there is a difference between prokaryotes and eukaryotes.
Is it like the starting codon?
Starting codon, it has nothing to do with this. Starting codon is going to be the same in bacteria and eukaryotes.
Okay.
So in bacteria, the ribosome, there is a site on the messenger RNA. We call it chain-Dalgarno sequence, and I have this in a slide in your presentation. Don't overlook this. It is important. What is a chain-Dalgarno sequence? Let me see here. I am positive that I have it somewhere. Here. Okay, so in bacteria, there is a sequence, we call it chain-Dalgarno, and this is like a very short nucleotide sequence. You know, maybe around 5 or 6 nucleotides, but there is a key feature for those nucleotides that they are rich in purine. Purines are adenine and guanine. So it's like 5 to 6 nucleotides, mainly purine, like adenine and guanine, before the start codon. And this is where the ribosome bind and start the process of translation in bacteria. In eukaryotes, there is no chain-Dalgarno. In eukaryotes, the messenger RNA has a cap. We call it 5' cap. And that 5' cap, this is where the ribosome bind and initiate the process of translation. Okay? There's a big difference between bacteria and eukaryote in this, how ribosome recognize the sequence. Okay? Let me see here. This is what I said. Remember, this is the cap. A cap is basically like it formed between two nucleotides, okay? And at the 5' end of the messenger RNA, it's between two guanine, okay? One of them is methylated, like this guanine here is methylated, it has a methyl group, and this is the guanine, which is at the 5' end. So there is a 5' to 5' interaction that will make like a cap, and at the beginning of the messenger RNA, and this is where ribosome recognize and start the process of translation. In bacteria, you don't have this. Viruses, they have this because they need their RNA to be translated, so they need to make it mimicking the host RNA, okay? Chapters gene expression is so important, and the difference between prokaryotes and eukaryotes, you need to be able to know the difference. you need to know that in prokaryotes, RNA is produced and no processing. So it can be translated directly. In eukaryotes, there is something called processing. And this slide is so important. It tells you that as soon as the transcript is formed, we call it primary RNA. It is not ready yet for translation unless some modification should happen. And those modifications are three. You need to know those. The first one is basically adding a cap. This is to modify the 5' end of the messenger RNA so it will be recognized by ribosomes. Number two, a polyadenylation tail is added. Poly A meaning it's a very long strand of like RNA made mainly of adenine. So it's a repetitive of adenine. We call it polyadenyl tail. So like a tail made of adenine mainly. And this is so important to stabilize the transcript. Number three, splicing. Splicing, meaning if you look at the primary transcript, you can see region in green and regions in orange. Those in green, we call those introns. Those in orange, we call those, or brown or whatever, we call those exon. Exon are the region that give amino acids. Introns has nothing to do with amino acids, so they need to be removed during the process. We call this splicing. Splicing meaning removal of introns and leaving only exons. So three things happen for a primary RNA transcript to give a mature RNA ready for translation. Addition of a cap, addition of a poly A tail, and splicing, meaning removing of introns, leaving only exons. This happens only in eukaryotes like human. In bacteria, none of this is happening. The RNA comes from the DNA and the process of translation happened immediately. So this slide is so important. You get by any chance, have you read that chapter? Guys, are you still there or I'm talking to myself now?
Yes.
You're here. We
Oh, you're
got
still
you on
there?
mute.
Because I can't see any, I'm focusing on the screen in front of me. I can't see the
I'm here.
window for Zoom, so I don't know who left and who stayed there. So now, this is very important chapter. Focus on this. Also, there is something that you need to know. You need to know the difference between a codon and anticodon on the transcript. Okay? So a messenger RNA will have the nucleotides, and the messenger RNA will form codon. Each 3 nucleotide will be responsible for the formation or the direction of one amino acid. We call this a codon. So a codon is 3 nucleotides on the messenger RNA that specify an amino acid. Also, there is something called anticodon. Anticodon, those are the complementary to the codon, and those are found on the transfer RNA. So the transfer RNA, that the uber or the left that carry the amino acid, has anticodon. The messenger RNA has codon. So the transfer RNA will know which amino acid need to come to this based on the complementarity between the anticodon on the transfer RNA and the codon on the messenger RNA. So if I ask you this, where you find the codons? You say, I found codons on the messenger RNA. Where you find anticodons? Anticodons are on the transfer RNA. Okay, and also remember that the direction of translation or making proteins is from 5' to 3'. So the ribosome read the messenger RNA from 5' to 3'. So now let's go to the big the most difficult part in this chapter, which is basically how to read and how to do transcription and translation for a piece of DNA. Okay. Let me see if I have. Okay, so first of all, let me show you this table here. And because you are going to have this table, you should have this table when you do your test, because I'm going to give you a piece of DNA and I will ask you to do translation. Okay. So let me do this. Insert the slide. Okay, let me do this. So if, say I give you a piece of DNA, okay? Like I will say in 5', G, 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 T, C, G, 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 C, E, A, A, T, T, C. Okay, here's a piece of DNA. Can you see this piece of DNA?
Yeah.
Great, and then let's do the complementary of it together. So when I say complementary, so I'm talking about the second strand now. So when I say the second strand, first thing I need to do is to change the polarity. So if the first strand is going from 5' to 3', the second strand is going to be opposite, from 3' to 5'. Now, look at the complement for G. What is the complement for G?
C.
C, C, C. What's the complement of T?
Is that A?
A.
Yeah.
What is the complement of C again? G. What is the complement of G? C, C, C. And then we have how many G's? 1, 2, 3, 4, 5, 6 G's. 1, 2, 3, 4, 5, 6 G's, right? 2, 4, 6, 7. 2, 4, 6, 7. And then I have another C. And then complement of S, T, 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 A, A, G. And then the 5. Okay? So let's do this. Now, if I give you a piece of DNA like that, and I said, okay, I'd like from you to do transcription, how to transcribe this, okay? I'm going to do transcription, and I need your help here. Transcription meaning creating messenger RNA, right? So now, if I give you these two strands of DNA, which one you will choose or select to make RNA? See this, let's first identify those two strands, because when you say transcription, you need to identify those strands. Okay? So you have the 5' to 3' strand, and you have another strand which is opposite. They have names. Okay. The one which is going from one of them, we call it the coding strand. And one of them, we call it the template strand. Okay? So which one is the template strand, you think? Template strand, meaning that is the strand of DNA where RNA polymerase will start making RNA from.
Is it the first line?
So the first one, this is where the RNA polymerase are going to copy and make RNA.
Yeah.
That will be a mistake, because when you make RNA, the RNA is going to be from 5' to 3', and you will never be able to make 5' to 3' using a strand of DNA which is in the same direction. So always remember anti-parallel. Always in DNA, RNA, always they are anti-parallel.
So it's like the mirror effect, right?
Exactly.
Okay.
So the template strand is going to be the strand where RNA polymerase will bind and start. It's the one that is going from 3' to 5'. Okay, this is the template. This is the one that you are going to focus on and make RNA out of it. This is the very first, very important step that you need to be able to determine first. So, and the other strand, we call it the coding strand. And I will tell you in a minute why we call it the coding strand. So now RNA polymerase is going to bind to this DNA, and it will use the template strand to make RNA. So now let's do together RNA, okay? Let's do RNA. So when I say RNA, okay, so the RNA is going to be from 5' to 3', okay? Now, RNA polymerase is going to recognize this and start making the complementary. So what will be the first nucleotide?
G.
3 Gs. And then A, what is the complementary of A?
Is it T, or is that the one that switches to U?
Absolute, very good. You're still awake. It is basically when you say, I say T because this is a complementary of A, but in RNA there is no thymine. It is only uracil. So every time you are going to see a complementary T, you need to change it to U. Never ever forget this. And then we have a G, so it would be C, and then we have 3 C's, 1, 2, 3, and then we have 7 G's. So we have 1, 2, 3, 4, 5, 6, 7, and then we have a C, so it's a G, and then we have 3 T's. What is the complementary of the T's here?
3 A's.
Perfect.
Oh, no. U's.
No, a complementary.
Oh.
The complementary.
Okay.
Yeah, only when you see the complementary of A, which is T, here you make it U. Okay? But
Okay.
here the complementary of T is A. It's okay, normal. Now, AA, what is the complementary of AA?
The UU.
Yeah, UU. I can't put TT here because it's UU. And then G is going to be a C. So that will be my messenger RNA. Okay? Control D. So that will be my messenger RNA. So this is the first step. So remember, a big chunk of your test will be a piece of DNA, and I will ask you to do transcription and translation. I need to make sure how to read this. If I give you a double-stranded DNA like this, you need to look for the strand which goes from 3' to 5'. This is your template. You need to select this and make your RNA out of it. Okay? If I give you one strand only, like 3' to 5', it's okay. You take it, I make your life easy. So you go ahead and make RNA directly. You don't need to do complement. Okay? What if I give you, I was mean, and I give you a piece of DNA, single strand, 5 to 3. So what you need to do first, you need to get the complementary, and then use the complementary to make your RNA. Okay? So look at the coding strand and the messenger RNA, and you will be surprised to see that they are exactly the same, except for any T is going to be a U. See? G, 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 G. Here is a T, it's a U, and then C, it's a C, 3 Gs, 3G, 7C, 7C, so on. This is why if I give you a double strand like this and I say, okay, go ahead and do transcription, you will be smart enough to copy the coding strand, okay, and every single T, you make it a U, and you get it. This is the most easiest way. So instead of doing the complement and be confused, if I give you a double strand like that and ask you to do transcription, you take the coding strand, which is going from 5' to 3', and you copy it exactly the same, but every single T, you are going to change it to U because there is no T on the messenger RNA. Clear?
Yes.
Any question for this? I will be more than happy to hear a question from you guys.
No.
Please, I beg you. If you have any question, ask me. Okay, now if I ask you to do translation, okay, and this is a fun part here. I ask you to convert this piece of RNA to protein. I will give you a table, and I prefer to use the wheel, okay? So let me take this wheel here. And I need to teach you how to read this. Are you familiar how to read this by any chance?
No, sir, not at all.
Good. So I'm glad to know this. So I
Me
need to
neither.
go. Okay, great. So now let me do this. I need to maybe move it. I need to move this. Okay. Okay, so look at this. So you have a piece of messenger RNA, and now you need to transform and make proteins coming out of this. Okay, so what you need to do to make your life easy, you take every 3 nucleotides and then see the corresponding amino acid. So this is the first 3 nucleotides here, 3Gs, okay? G, and now I need to see what kind of amino acid will come from this 3Gs here, okay? So look at the wheel here. And if you look at the wheel clearly, you will see like there are 3 circles. An inner one, a middle one, and then an outer one, okay? 3. So, and when you read from the middle or the center is going to be the 5' end. This is where to start reading. 5' end to 3' end. So reading from the inner circle first, then the middle, then the outer. Your direction for reading from the middle to the, sorry, from the inner to the middle to the outer. So if I read G, 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 so the middle, the inner one here, this is G. So this is my first G. Then I will move to the middle one. Then is the middle one. Where is the second G? Here. So G, G, and then I will move to the outer one. Where is the G? Here. So G, G, G. What is the amino acid? Glycine. Glycine. Okay. So I will do this, and I will add glycine. Now, take the second codon, UCG. UCG, again, go from the inner, U, C, G, U, C, G. What is the amino acid? What is the amino acid?
So serine.
Serine, perfect. This is serine. Now the third codon, GGC, and I need one of you to tell me the amino acid for GGC. GGC, what do you think will be the amino acid?
It's the same as the first one.
Yes, it's a glycine. GGC, it's a glycine. Now move to the fourth one, 3 C's. I need somebody else to tell me what is the 3 C's, what is the amino acid that can come from this? 3 C's, huh?
Is that protein?
Proline, yes, proline, yes.
Proline,
C is a proline. Proline, perfect. Great. Make this proline here. And then another 3 C's, right? So that will be another proline. Okay. Now GAA, what is GAA stands for?
Is that be glucose? I don't know.
Yeah, that's okay. You don't need
Okay.
to. I'm not going to ask you, to tell me what is the name of the amino acid, but yeah, you need that. At least I will tell you in a minute what is this 3 letters, because there is something else. I don't need to confuse you at this moment. But a GAA, again, it is a GAA. So this is not glycine, right? It's different. It is GLU, and GLU means glutamic acid. Okay.
Okay.
So GU, glutamic acid, is GLU, okay, or glutamate. And then AUU. AUU.
It's the LLE.
Yeah, it is isoleucine. ILE, which is
Oh, it's the I,
isoleucine.
okay.
Yeah, it's the first letter is cap. Okay, capital letter. So it is isoleucine. Great. So now you know the idea. Now, if one nucleotides like this left, you leave it alone. You don't need to mess with it, because it's not any amino acid, because you need to have three nucleotides to be able to tell the amino acid, right? So if it finished with a C like this, leave it alone. So you have those amino acids. This is a translation here. And if I ask you to, we call this the 3-letter code for amino acid. So amino acids can be placed like with the first 3 letters. Like here, glycine, we have GLI, serine, we have SCR, okay, proline, we have PRO, or there is something called 1-letter amino acid, 1 letter only, okay? So for example, alanine is an A, arginine is an R, okay? So if I ask you, after you do translation, to put it in 1-letter code for amino acids, you need to change each one of those to only 1 letter. So if you look at glycine, you go here and you see that glycine is a G, so you put G. If I say glutamic acid, like here, GLU, you go to the table, and GLU is an E, so to put an E, because I will ask you, I will put like a code, and I will ask you to solve the puzzle for me and give me the code. We're using 1 letter only. Simple, and it will be basically lots of point in this question. When I give you a piece of DNA and ask you to do transcription and translation, you need to follow up with this. It is so important. Okay. One last thing I need from you to see, some amino acids can be produced using different kind of codons. Like for example, look at serine. How many codons for serine? UCA, UCG, UCU, UCC. 4 different codons can code for serine. Okay. Similarly, glycine. You have GGG is a glycine, GGC is a glycine, GGU is a glycine. Okay. Also, what I need from you to know from this, there are some codon that if the ribosome will meet, it will finish and terminate the translation. We call those stop codon. And there are 3 stop codons, and all start with the letter U. You have UGA, UGA. If you see a UGA here, it means that there is no amino acid, and you will stop there. UGA is a stop codon, UAG is a stop codon, UAA is a stop codon. So 3 stop codons all start with the U letter, uracil. So if the ribosome will see a UAA, it says, okay, enough. I'm going to take some rest. There is no need for more translation. I will stop here. We call those the stop codon. That is the signaling for stopping translation. Okay. I hope you got this and you understand something today. And I will be more than happy to hear if you have any question for me.
Is this on the Canvas PowerPoint?
Yes, yes. That was placed last week.
Okay.
Yes.
When
Okay.
is the test?
The test is going to be next week.
Okay.
I'm telling you, I will give you the date exactly, but I will send you an announcement for the date. But it's going to be next week.
Okay.
This week you are going to be busy with the practical.
Oh yeah, was the pop quiz that's coming up in a few days? So that's over chapters 16 and 17?
Yes.
Okay.
Yes. Okay, any question for me?
Nope.
No.
Okay, you can, what I'm going to do, I'm going to hopefully be able to put this in, I send you a link for this. So you will have it also. So if I missed you at some point, hopefully you will be able to get back to it. Okay, so okay, good luck, and don't hesitate to email me if you have any question, please.
Okay.
Okay,
Yes, sir.
perfect, great. Thank you very, very much, guys.
Okay.
You guys have a great day and be
You too.
safe and healthy.
Yeah.
Okay, thank you. Bye-bye.
Bye-bye.